Welcome, medical students, to today's lecture on the composition and organization of the extracellular matrix, ECM. The ECM is a complex network of macromolecules that provides structural support and regulates cellular behavior in tissues throughout the body. Understanding the composition and organization of the ECM is crucial for comprehending various cellular processes and their implications in health and disease. The ECM is primarily composed of structural proteins, proteoglycans, and glycosaminoglycans, GAGs. These molecules interact and form a highly organized framework that supports cellular function. In addition to providing structural support, the ECM also stores growth factors and cytokines, regulating their availability to cells. Collagen is the most abundant structural protein in the ECM. It forms fibrils and provides tensile strength to tissues. Another important structural protein is elastin, which confers elasticity to tissues such as skin and blood vessels. Fibronectin and laminin are adhesive glycoproteins that facilitate cell attachment and migration within the ECM. Collagen, as mentioned earlier, is a crucial component of the ECM. It is synthesized by fibroblasts and secreted into the extracellular space. Depending on the tissue type, different types of collagen, such as types 1, 2, and 3, are present. Each type has distinct structural properties, enabling tissues to withstand different mechanical stresses. Elastin, on the other hand, is responsible for tissue elasticity. It is synthesized by fibroblasts and smooth muscle cells. Elastin fibers can stretch and recoil, allowing tissues to return to their original shape after deformation. Proteoglycans are complex molecules consisting of a core protein with covalently attached glycosaminoglycan, GAG, chains. GAGs are long, linear polysaccharides composed of repeating disaccharide units. The major GAGs found in the ECM include hyaluronic acid, chondroitin sulfate, heparin sulfate, and keratan sulfate. Proteoglycans and GAGs contribute to the hydrodynamic properties of the ECM and help maintain tissue hydration. They also regulate the diffusion of signaling molecules and growth factors within tissues. Cells interact with the ECM through specific cell surface receptors. The most well-known receptors are integrins, which are transmembrane proteins that bind to ECM components such as fibronectin and collagen. These interactions provide structural stability and regulate signaling pathways within cells. Integrins mediate bidirectional signaling, transmitting signals from the ECM into the cell and vice versa. They regulate cell adhesion, migration, proliferation, and differentiation. Dysregulation of integrin signaling has been implicated in various diseases, including cancer and fibrosis. Cell adhesion complexes are multiprotein assemblies that link the ECM to the cell cytoskeleton. Focal adhesions and hemidesmosomes are two types of cell adhesion complexes. Focal adhesions are mainly involved in cell migration and mechanotransduction, whereas hemidesmosomes provide stability to epithelial tissues. Focal adhesions are composed of integrins, linker proteins such as talin and vinculin, and signaling molecules. They play a crucial role in coordinating the dynamic changes in cell shape and motility. The ECM is a dynamic structure that undergoes constant remodeling. Several enzymes, such as matrix metalloproteinases, MMPs, are responsible for degrading and remodeling ECM components. This remodeling process is critical during tissue development, wound healing, and tissue repair. MMPs cleave ECM proteins, allowing cells to migrate through the newly remodeled tissue. Failure of ECM remodeling can lead to aberrant tissue architecture and impaired wound healing. During embryonic development, the ECM provides structural guidance and signals that regulate cell behavior. It helps determine tissue boundaries, shape organs, and orchestrate complex morphogenetic processes like organogenesis. The ECM acts as a scaffold for cell migration and differentiation, contributing to tissue organization and proper function. Stem cells reside in specialized microenvironments called stem cell niches. The ECM plays a crucial role in regulating stem cell fate decisions, including self-renewal and differentiation. It provides physical support and supplies signaling molecules that control stem cell behavior. Cell migration and invasion are essential processes for tissue remodeling and immune responses. The ECM acts as a physical barrier and provides guidance cues for cells to migrate or invade specific tissues. Cell-ECM interactions, mediated by integrins and other adhesion molecules, are critical for directing cell movement and determining tissue tropism. The ECM not only provides structural support but also transmits mechanical signals to cells. This process, known as mechanotransduction, involves the conversion of mechanical forces into biochemical signals that regulate cell behavior. For example, stretching of the ECM can activate intracellular signaling pathways, influencing cell proliferation, differentiation, and gene expression. The ECM plays a central role in wound healing and tissue repair processes. After injury, various cells, including fibroblasts, migrate to the wound site and produce new ECM components. This newly formed ECM provides a provisional scaffold that guides cell migration, promotes angiogenesis, and ultimately restores tissue integrity. Alterations in ECM composition and organization are associated with numerous pathological conditions. For instance, excessive deposition of ECM components, as seen in fibrosis, can disrupt tissue architecture and impair organ function. Dysregulation of ECM remodeling processes has also been implicated in cancer progression and metastasis. In conclusion, the ECM is a complex network of macromolecules essential for tissue development, homeostasis, and disease pathogenesis. Its composition and organization, including structural proteins, proteoglycans, and cell adhesion molecules, dictate cellular behavior and tissue function. Understanding the intricate interactions between cells and the ECM is crucial for unraveling the mechanisms underlying various physiological and pathological processes.